Wednesday Night Lab, we do this every Wednesday night, 50 times a year. So for those of you who are new, come on back often. And then I want to introduce Jaime Cordova to you. Jaime has been one of the lead organizers of Darwin Days this year. He's been a great person to work with. And he also gave a wonderful Wednesday Night Lab talk last summer about the 50th anniversary of Apollo 11. Not bad for a geneticist. So, Jaime, take it away. Thank you.
Thank you. So hello, everyone. Thank you all for coming to tonight's Wednesday Night at the Lab in celebration of Darwin's birthday. First, I'd like to start off by thanking Dr. David Baum and the Wisconsin Brewing Company for providing some of the drinks that you all enjoyed during our reception earlier. And again, thank you all for coming in celebration. I also want to plug that if you want to learn more about Dr. Orive's work, we will also be hosting another talk tomorrow at 12:30, and you can learn about that at the Crow Institute website, evolution.wisc.edu. But now it is my pleasure to welcome Dr. Maria Orive from the University of Kansas. She's a professor of ecology and evolutionary biology. She was born, I'm going to steal Tom's thunder. So she was born in Havana, Cuba. She went to high school at North Thurston High School in Olympia, Washington. She did undergrad at Stanford University in California. Woohoo, California. She did her PhD at UC Berkeley, also woohoo, California. And then she did her postdoc at the University of Georgia, and she actually also did a second postdoc at the University of Edinburgh. So with that, I'd like to welcome Dr. Maria Orive. Thank you.
Well, I wanted to say thank you to the organizing committee and especially to Jaime for inviting me. It's been a lovely visit so far, and I'm really looking forward to my day tomorrow visiting again. It's Darwin's birthday, which is wonderful enough as it is, but I was so thrilled to be invited to the Crow Institute. So when I was a graduate student at UC Berkeley, go California, I went to a math biology workshop that was over at Stanford. And at breakfast, I met a man who talked to me for a while, and his name was James Crow. And I realized after I talked to him for a few minutes that he was actually the famous James Crow. And I told him all about my nascent PhD project, and hope, so we talked over breakfast. And about a year later, I was at an evolution meeting. I can't remember where it was that year. And I saw him, and I was like, I'm going to go say hi. He's not going to remember me, but I'm going to go say hi. I'm going to be brave. So I walked up, and I said, "Hi, Professor Crow, you don't remember me?" And he says, "I have forgotten your name. But how's that project going? You were working on effective population size using a nice coalescent model. Let's talk about it." And I think my feet didn't touch the ground for the rest of the meeting. So that's my memory of James Crow, somebody who was really interested in what students were doing. Also, the man who wrote literally the textbook that I used. So it's a real honor to be here at the Crow Institute. It's amazing. And there's a little little museum thing out there where you can see some of his papers and photographs, and I was really happy to see those. All right, so I decided to talk about what I do, which is use mathematics to think about biology. And so if you Google mathematics and biology, you find, it depends on what your search safety is, but you find all sorts of interesting things. You might find stuff like this. So you might find humorous cartoons implying that people who do math basically can't explain anything to anybody. But then you might see heartbreaking things like this. Majors you won't need much math for. And in this thing is biology. And it's like, ooh, sad. But I think everyone in this room knows that that's not true, right? Biology has a long history with mathematics, especially ecology and evolutionary biology. Certainly at the Crow Institute, of all places, that's definitely a known thing. So here's just an example. This is from Sally Otto and Troy Day's wonderful book, A Biologist's Guide to Mathematical Modeling, I think is the title. And they just said, okay, for a particular year, let's look at 3 big journals in ecology and evolution and ask, well, how much math is there? So the AmNat, Ecology, Evolution, some of the well-known journals. And you'll notice that if we say using models generally and include things like analysis of variance or regression, statistical models, it's everything. which shouldn't surprise anybody. Okay, that's fine. But even if we're much more specific and say, what about mathematical models being used to obtain the results, and even equations actually present in the paper? It's a big proportion. So, it's a long history, and this is a little older now, but I don't think the numbers would be very different if we did it today. So I wanted to say a little bit about how we use models to make sense of biology. And there's lots of ways to divide things up, and people like to divide things into groups of 2 for whatever reason. So here's 2 groups. We can use models to explain what we do see. So for specific tests of hypotheses. And I'm going to give you guys an example that I'm fond of that is about how we use models to look at the dynamics of HIV infection within a human host that's infected. But we also use models to predict kind of what we might see, to generate hypotheses. And this has been incredibly important in ecology and evolutionary biology. And I'm going to give you an example from my own work here, looking at what are called evolutionary lag and rescue in organisms that have complicated life histories. So I'm going to talk a little bit about each of these. So here's some data. This is something that we do see. So this is data from a study from the mid-90s showing the time course of infection within a single individual that eventually died of AIDS. And so 2 things are being tracked over time. And you notice the scale changes from weeks to years later. We're tracking the number of CD4 plus T lymphocytes per, I think, cubic millimeter of blood. And that's this solid line. So we see this sharp decline, but then a recovery, and eventually a slow loss until death. And if we follow instead the viral load, which is how much HIV RNA is in the plasma, we see this spike, and then this down, and then this slow increase. And there's these dynamics of up and down here that it would be interesting to be able to understand why does that happen? And one way we can ask is to create a model. So the kinds of models that I mostly work with, turns out I lit those up. So that's the blue one is the cells, and the red one, I guess, is the virus. Okay. I tend to use models that explain dynamical systems. So systems that change over time, just like what I just showed you there, right? A change in the number of cells or the amount of virus over time. Mathematicians have lots of ways of describing these. Sometimes we use things like differential equations. They describe the rate at which a variable changes over time, and these are continuous in time. We often use this kind of notation, right? dn. So n is a variable we're following over time, t, and it changes at some rate, as some function. And then sometimes we use recursion equations that describe the value of a variable in the next time step that are discrete in time. The notation sometimes looks like this. N, the variable at t plus 1, is a function of what it was at time t. Or sometimes we get really shorthandy and we write n prime is a function of n, with the idea that n prime is in the next generation. And in all these kinds of models, what we're doing is we're following something over time, and we're seeing if we can have a reasonable model of that. So let's think about that. And for the virus example, we're going to use differential equations. So those spikes up and down immediately made people who model biological systems think of other kinds of systems that have similar spikes up and down. And this is probably one of the most famous data sets. And I want to get the name of the person right, so I'm going to look and memorize things. So this is data that was analyzed by a biologist named Charles Gordon Hewitt in 1921. And what it is data from fur trappers turning in pelts to the Hudson Fur Company for lynx, that's the predator, and snowshoe hares. And the hares are in the solid, and the dashed lines are the lynx. And you see this up-down pattern that tends to go sort of slightly asynchronously. And this was, you can tell that the data started in the 1850s and goes on and on. And you can explain the dynamics of this relatively easy with a simple, what's called a predator-prey model, which are a set of differential equations. So here's probably the simplest version. There's many more complex versions, but here's the simplest version of the predator-prey model. So we keep track of the number of prey, that's x, the number of predator, that's y, and we set up this model. And at first you look at it and you're like, oh, that's a bit complicated. It's actually pretty easy to break down. if we think about the prey, we think about ways that the number of prey increase and ways that the number of prey decrease. So for the growth rate of the prey, the hares, we make a simple assumption. We say it's constant, and it's what we call a per capita. So we multiply that rate by the number of hares that are sitting around. So we're assuming they're reproducing at some constant rate that depends on the number of hares that are there. That's a simple way to look at it. And then how do they decrease in number? Well, they decrease in number when they get eaten. Yeah, well, they'd probably decrease in other things, but we're saying that's the main thing that's changing the numbers. Okay, so if that's true, then we have to have some sort of capture rate, the death rate of the prey. And this is what we call a mass action term. A mass action term basically says the rate at which this thing happens depends on either the number or the density of two things coming together. So they have to bump together randomly. So we're assuming that when a hare meets a lynx, they occasionally end up with the hare being eaten by the lynx. And this is that capture rate. So increasing and decreasing. What about the lynx themselves? They also have an increase and a decrease. They have a growth rate that's related to that same mass action term, right? They have a predator growth rate, and it's kind of a conversion rate. You can think of it as the rate at which you convert eaten hares into baby lynxes, right? It's an energy conversion rate. So that's what that is. So that's a growth rate here. It was a decrease in the other guys, but it grows here. And then, of course, the lynx also die. And in this very simple model, the death rate of the predator is completely independent of the hares. The hares aren't doing anything. And once you put that together, you get a model that mimics those ups and downs incredibly well. It's like, you can make it more complicated, and of course the reality is more complex than this, but it actually predicts really well that long range of data from the Hudson Bay Company. So what does that have to do with viruses? Again, ups and downs. So let's think about explaining like the viral load. What is this number? And how can we explain how it changes? In 1996, Phillips published a model, probably one of the first models, but there have been many models of HIV since then, and even around that time. But this is a relatively simple model, where he used the same sort of differential equation predator-prey system to explain what's happening inside an infected host. So he said, we're going to track four things here. The number of susceptible CD4 plus cells, those are the cells that can get infected by HIV. The ones that are latently infected, that's the ones that have HIV in them, but the virus isn't replicating. And then the actively infected ones are the cells where the virus is actually replicating. And then also the number of virus particles. And the key here is to realize that in this model, the virus is the predator. And it interacts with the cells, the susceptible cells, as prey. And the dynamics are actually very, very similar. And you can put together a predator-prey model that mimics the dynamics of infection inside the host, at least for part of the system. So I'm going to put together this model in the same way that we did the lynx and the hare model. So just to remind us, this trace, this data here, is the CD4 T cell count. It groups together all cells, because you can't, when you're doing this kind of study, they're not actually knowing which ones are infected and which aren't. So really, this trace is these three differential equations. And this trace is this differential equation. So the question is, can we capture that behavior? So what we're going to do is have a little flow diagram, and we're going to keep track of those four things, right? The susceptible cells, latent cells, actively infected, and those virus particles. And the whole model is complicated. But if we break it down the same way we did for the lynx and the hare, you guys will think, it's not that complicated. I could do this. I've done this already. Come on. Do it at breakfast. Okay. So here's our susceptible group. And the first thing is we need input into that group. This is a key assumption of the model that I'm going to come back to later. And it's that the input of susceptible cells, these guys come from the immune system, and that it's some constant. So this gamma function, it's constant per unit time. We'll come back to why that assumption is not completely true and when it breaks down. But we assume that they're just coming into the system from the immune system. Okay, so how do susceptible cells leave? Well, they can die. There's some natural death rate of these cells. We'll call it mu, and it's a per capita death rate. So we've seen per capita things before, right? That makes sense. So that's one thing, it can die. But the other thing is, what's the other way that we can lose a susceptible cell? It gets infected. So here's the infection. It either becomes latently or actively infected. For now, we're not going to worry about that. And what do we see here? A mass action term. So it's the same sort of thing. The predator needs to bump into the prey, and then there's some infection rate. So this is exactly analogous to that term that we did with the lynx and the hare. So this is our infection rate, the number of virions floating around, and the number of susceptible cells. And what we're going to say is there's some probability that once you get infected, that you become latently affected or actively infected. We'll call the probability P for latently infected and 1 minus P the other way. So that's it for the susceptible cells. Here's the latently infected cells. The input there is just that mass action term multiplied by P, the probability you go this way instead of this way, right? So we assume that that's some probability that you're latent. The latently infected cells can die. And because they're latently infected, the HIV is not active at all in those cells. So we're going to assume initially that the death rate's the same as for the uninfected cells. As far as that cell knows, there's no virus in there. And then, of course, there's another way latently infected cells can leave the group, and that's becoming active. So there's some conversion rate from latent to active that happens, taking latent cells into this active pool. We can do the same sort of thing for the active pool. Here's that mass action term again. Now we're going to multiply it by 1 minus P. That's the probability you go this way and end up in an actively infected cell. So that's the input. And then we're going to have a couple of but we have one more input. This one, right? This appeared as an output of latent, but it's an input into the actively infected. So this is the conversion from latent to active. And then we have an output, which is the death rate of actively infected cells. Now these guys are actively budding virus. They're definitely not the same as cells that aren't actively budding virus. They're sick. And so those cells die at a much faster rate. They have a different rate. There's a dashed line here. And we're going to see that actively infected cells make virions. But the reason it's a dashed line is an actively infected cell that sheds virions doesn't stop being actively infected. So this is an input into this, but it's not an output, because actively infected cells stay actively infected even when they bud off a virus. So finally, here's our virions. The input is, again, from these actively infected cells, although that doesn't leave these guys, but it's some rate pi, the budding rate of virus particles from infected cells, multiplied by the number of infected cells, actively infected cells. The virions themselves are cleared from the plasma at some rate, some death rate. That's per capita. And of course, they get taken up by that mass action term as well. Now, it turns out that compared to this input and this output, this is actually a tiny proportion of the virus, because you just need one virus particle to infect a new cell. But we leave it for completion. Usually when they analyze these actual models, they ignore that term in there. And then you've put together the whole thing. So what happens when you put that together? Well, you can use some estimates from the literature and say, well, does it predict what we expect to see? So the time scale is not quite the same here. But you see this peak and decline is exactly the same sort of peak and decline that you see here. And I didn't show the cells, but the cells also go down and back up again. So that initial behavior of the system, where the virions go up and the cells go down and then they switch, that behavior is completely explained by a predator-prey model of just the virus sort of using up a lot of the susceptible cells, like the lynx initially using up a bunch of the snowshoe hares. And so the interesting thing is, you might, if you had seen this, imagine that you saw this behavior in a patient that you'd given a new drug to. You would see a decrease. You'd see a spike in the virions, but then a decrease. And you'd see a recovery in CD4, and you might say, hey, that drug's working. So it's really important in all things to understand what the system is actually doing even without any perturbation, right? So this is an important null model for what happens of the virus in the body. All right, so that was my example of explaining what we do see, a specific test of a hypothesis, that behavior that we see in the virions and in those cells can actually be explained by a really kind of simple process, a predator-prey process. So now I'm going to talk a little bit about predicting what we might see when we try to generate some hypotheses. And this is some stuff from my lab. So one of the questions that I think all evolutionary biologists are interested in is, how do organisms track environmental change? So we've got kind of the poster child for environmental change here, the peppered moth. So here's a peppered moth on a tree somewhere in England, matching its background beautifully. So if we think of a trait being the amount of pigment in its wings or something like that, and we'll call that trait Z, whatever that trait is in this moth, it's at the spot that maximizes fitness, right? That maximizes its probability of like evading being eaten. So that moth has, let's say, has great fitness. It's sitting right where it should be. It's awesome. But you know, the world changes and the environment changes and the industrial revolution happens and we have lots and lots of soot on our trees. And now my friend, who has the same mean phenotype, finds itself really far from the optimum, where now the optimum might be really dark wings, right? I should have really dark wings, but I don't. I'm sitting here. This is not good. So I'm quite a ways from the optimum, and now my fitness is much lower than it should be. Birds are eating me all the time. So what can I do? Well, I can move. I can go somewhere where there's no pollution. That's one thing. And certainly many times organisms do that. I can go extinct. That kind of sucks. That's not good. I can adapt, right? If natural selection has a way to move me to this new optimum, I could adapt and follow that optimum. So that's the process that I'm really interested in. And I'm especially interested in how do different types of reproduction affect that process. So this term, I used it in, I've used it already without defining it. It's evolutionary lag, sometimes called lag load. It's just basically the difference, it's an abstract idea of the difference between the phenotype trait mean, the way it is right now, and what that optimum should be. So here's one type of lag, just the difference between Z bar and the optimum theta. Here's Maynard-Smith's version, which is the scaled version, which I'll use in a fitness function later. And the reason we're interested in this idea of evolutionary lag is we expect to see greater evolutionary lag when environmental change is fast. So when things like human perturbations happen, or when there's changes to the climate, or when there's invasion of new species. So we're especially interested in those kinds of situations. So this also brings up another term, this idea of evolutionary rescue, which brings into play sort of eco-evolutionary dynamics. So if we imagine we have a population that's sitting around, and then there's an environmental change and fitness suddenly goes down, we might see demographic decline, right? The population size goes down. Now, if natural selection is able to move that population closer to that new optimum, we might see what's called evolutionary rescue. We might see the population be able to adapt and increase in size again. That would be great. But people who are thinking, but what about happens here, right? The population's really small. And small populations are vulnerable to demographic chance. They're very vulnerable to extinction. And so there's kind of this trade-off, right? How far down can you go before you can't get rescued? So this is a question that we'd like to be able to predict. So the kinds of creatures I'm especially interested in are organisms that have complex life histories that include stage structure and that reproduce asexually or clonally. I've got some examples here of some favorites. This is an invasive plant on a beach in Hawaii where it's warm. I'm just looking at that going, a beach in Hawaii. It's called Vitex rotundifolia. And it is spreading. It's hard, you can kind of see. It's spreading via runners, right? And those runners sometimes break off and leave the little plantlets isolated, and sometimes they stay hooked together, but this thing is spreading all over this beach. And here is a staghorn coral. This is actually the remnants of a fragmented staghorn coral. So it's now a whole bunch of colonies that used to be like more than one. There used to be, I think, one colony. So this is a coral that's fragmenting. And so those fragments are genetically identical to each other. That coral can also reproduce sexually. It also produces gametes. So does this guy. This reproduces sexually too. Here's Hydra. Hydra's like the poster child for asexual reproduction because it's beautiful, right? I mean, that's attractive. Here's mom, and here's the budding Hydra coming off her somatic tissues, and that is a genetically identical little clone, right? So that's pretty cool. And this is Poa bulbosa. This is a grass, and it's producing. It looks like it's making seeds, but it's not. It's making bulbules. It's making asexual propagules that are somatic tissue also. So those are also genetically identical to the mom. And I'm especially interested in these types of organisms because there's a lot of evidence that invasive spread is sometimes associated with clonal reproduction. So this looks like a field, right? It looks like, oh, I'll go for a walk on that. You wouldn't because it's a body of water. And it's covered completely by this is an aquatic fern called Salvinia molesta. And just the name, molesta, tells you that whoever named it didn't like it very much. These are little tiny things, and it's spreading vegetatively. It's covering that entire field. And we have lots of examples of sort of the clone taking over kind of thing. So this is the question that we've been asking is, how do plants that have sort of complex stage structures and animals that have complex stage structure and clonal reproduction affect a population's ability to track environmental change and maybe have evolutionary rescue? So to do that, we need a model to track things that are changing in time. Remember, I said I'd do dynamic models. I'm taking it back. Dynamic models of things that change in time. Here, the thing that's changing in time is that phenotype, that trait, right? We want to find out how that trait can change. We use a kind of classic quantitative genetics model that says, hey, we've got a phenotypic trait, and we're going to keep track of additive genetic factors and also non-additive genetic factors and maybe random environmental factors. So what do I mean by that? There's a big difference between sexual and clonal or asexual reproduction on how the genetic variation that underlies traits is inherited. So this is my little cartoon that I use for this. If we imagine a diploid organism, so you have two copies, one from mom, one from dad, that has sexual reproduction in a random mating population. I need it to be a random mating population. Then Mendelian segregation at a locus and recombination between a locus means that generally, on average, the only thing that is inherited by offspring of this quantitative trait would be what we call the additive allelic effect. All right? But if instead of reproducing that way, you are a diploid who reproduces clonally and makes a copy of your entire diploid-linked genotype, then you're also passing on any dominance interactions between alleles at a single locus and any what we call epistatic interactions, alleles across different loci. So these guys get passed on as well. So we have this, that's what I meant by non-additive genetic factors, these dominance and epistatic factors. And then also, if there are aspects of the environment, of random environmental components of the phenotype, if those are passed on with somatic tissue that's formed during development, it's possible that the clonal offspring will have that as well. So we define this parameter that we called rho, that's an association between this E, which is those non-additive genetic components, and the environmental factors in a parent and its clonal offspring. This rho is 0 under sexual reproduction in a random mating population, under usual assumptions. And we might imagine it's close to 0 for types of asexual reproduction that look a lot like sexual reproduction. This is not from my yard, but it could be, right? This is our favorite asexually reproducing dandelion. But think about types of clonal reproduction where the offspring takes up a lot of tissue from the parent, and where the phenotype might be something that's already developmentally sort of stabilized in that parent. It might be closer to 1 for something like this. So the type of clonal reproduction will affect this. All right. So I said I was going to do recursions. I am. So we need to follow the change in something. I've been saying we do dynamic models. What are we going to follow in this model? We're not going to follow the number of virus. What are we going to follow? We're going to follow numbers. Numbers are nice. The number of individuals for each stage in the life history, and I'll define that a little bit better in a second. And then we're going to follow the mean genotype and the mean phenotype, because we want to know how phenotype follows adaptation. So again, Dynamical models describe systems that change over time. Now I'm going to do this. I'm going to use recursion equations. And I'm going to use a model that's discrete in time. It's not discrete in everything, but it's discrete in time. And I'm going to use actually both of these notations, because I wasn't consistent when I wrote this. So when I talk about life history structure, one way to describe a life history of an organism is by something called a life history graph. So we're often thinking about age-structured populations. Humans are sometimes used that way when we talk about the demography of a human population. Here's a completely made-up example of like some line drawings I found, that were common use, so I could copy them. So here we have some fish that I've made bigger artificially, and some little, fish zygotes. And we might imagine that we have an organism that has this life history, that we have sexually produced individuals of stage 1, and then after a year we have stage 2 individuals, and after another year stage 3, and after another year stage 4, and maybe nobody lasts longer than that. And the possibilities for moving around in this life history is surviving from one stage to the next, from one age group to the next. And then we have these arrows showing reproduction, so production of these guys. And that's kind of a typical life history. And we can sort of summarize the details of that using a matrix model. It's sort of projection matrix that gives us the number of individuals. Here we're using that prime notation. The number of individuals in stages 1, 2, 3, and 4, the next time step, as a function of them in the previous time step. And all that matrix does is give us a way to put them together. So these are the survivorships, and these are the fecundities. So here we have the number of individuals in stage 1. Those are those zygotes. And the stage 1 individuals don't contribute to themselves because they don't reproduce. But these guys all do. And so the number of individuals in stage 1 in the next time step is the number of individuals that were in stage 2 multiplied by their fecundity, plus the number of individuals that were in stage 3 multiplied by their fecundity, plus the number of individuals that were in stage 4 multiplied by their fecundity. So if we add that all up, we get the number of these guys. That makes sense. And we can do the same thing for all the other stages. The only way to get to this stage is to survive from stage 1. There's no other. Everything else is a 0. So the number of individuals in stage 2 is just the number of individuals that were in stage 1 that survived. And the other 2 steps are exactly the same, right? Because the only way to get to 3 is from 2, and the only way to get to 4 is from 3. So we've made recursions, simple recursions. Now the types of life histories I'm interested in are a little bit more complicated. And you can tell I'm not a real biologist. It's like, look, I drew something. I didn't draw these. So here's a little bit more complicated life history. But let's just pretend that this cartoon is something realistic. So we again have sexually produced, this is a little planula larvae here. Little sexually produced individuals. You only get here via sexual reproduction. And then you have a few other stages. Maybe a single little zooid, maybe a couple of buds of a zooid, and then maybe a coral colony or something like that, right? And now you have different kinds of movements, and these stages are no longer ages because there's more than one way to get there. You could stay in the same place. That's what makes them a stage instead of an age. You could go backwards. Maybe you fragmented like that coral colony. Or you got eaten partially and you got smaller. You can get bigger. So there's a few different ways. And so we're going to break up those transitions, but we're going to do the exact same thing. We can use a model that projects how you get into each of those stages from the previous time step. And now there's 3 different kinds of entries in there. There's what I'm calling simple transitions. So that's just a movement from one stage to another. I was here and then I grew to here. Or I just stayed in this stage from one time unit to the next. Or I got smaller. I can get bigger, I got smaller, I can stay in the same place. Some of these could be 0, so maybe they're not possible. Then sexual reproduction, I'm always going to just define one of these as sexual reproduction, so I can always loop those into there. But then I might also have a type of clonal reproduction. So maybe it's possible to create that one little zooid by budding, by asexual budding. So there are transitions. And notice that there's 2 ways to get to this transition, right? You can shrink or you can bud off clonally. So we're going to keep those separate. We put all those together, we do a lot of math, and we get recursions. We get the number of individuals in stage I, and it's a function of the ones that came from transitions, the ones that came from sex, fecundity, and the ones that came from clonal reproduction. So we're just tracking everybody. So that's great. That's how I did the recursion for the numbers of individuals. But I also said I was going to track the genotype and the phenotype. And here I'm going to have to wave hands a little bit, because now things are going to get a little bit hard to explain, and you're just going to trust me. So I'm going to write a recursion equation for genotype and phenotype. And it's discrete in time, but phenotype and genotype are continuous. So it's discrete in time, but it's continuous in genotypic and phenotypic space. That's not so great. But nevertheless, it looks kind of like the things that we've done before. It's a recursion, a value of this given a value before. Now I'm going to have something complicated that I'm not going to define very well. But it's basically the distribution of genotype and phenotype and a function that tells me how that changes. So the best I could do is to make a little cartoon. So we imagine that genotype and phenotype for a given stage have some distribution. So we might imagine that for a given stage, the phenotype can take a lot of different values with different probabilities. Like it's most likely here, here's the mean, but there's other possibilities. And here's the genotype. And what I'm saying is I have to develop a mathematical model that does a projection, not of a value of one thing, but of a distribution. And so that's what I'm basically doing, is something that says, what is this distribution as a function of this one? And then what is this distribution as a function of this one? And the actual reality is that this distribution is a function of both of these, right? And the same is true. But that's basically what we're doing. We're doing a projection, same as before, but instead of a single value, we're doing a distribution. I wanted to show you a little bit of how we did it. The cool thing is we were able to do the recursion by breaking it up into those three things: transitions, sexual reproduction, and clonal reproduction. This thing is just the number of new stage I individuals that happened from transitions. So this proportion is, these are all the new individuals in stage I, and this is how many of them were from transitions. So this is the probability that you came from a transition. Okay, so with this probability you came from a transition, sex, clones. And then this thing gives that distribution. This one's the easiest one to do because it just uses that transition matrix to project where you came from. The other two are a little bit uglier. So here's the one for sex. And for this one, this function predicts what the genotype and phenotype is going to be of sexual offspring. To do that, I have to take things into account like Mendel. I have to take into account alleles and mutation, right? And selection. So that's where all the sort of real pop gen happens and the real quantitative genetics happens. But it's not too bad. It's in the appendix. All right, so that happens there. And then these are the proportion that are clonal. Here's the distribution for the clonals. Now you might think, well, that's easy, Maria. They're clones. The genotype should be exactly the same. I am allowing them to have mutations, though. So I do allow somatic mutations. So there is some movement of the genotype, a little bit of movement. I just say that it's not directional, so it's random. All right, so we do all that, and we get this. Yay! You're like, okay, z bar prime as a function of z bar in the previous thing, g bar prime. Yeah, okay, that sort of makes sense. What if we took it and made it a little simpler? So let's take something much simpler. Let's take a very simple life history. The simplest life history I can give that has both clonal and sexual reproduction. It doesn't have any stage structure. So we have a single stage. We have some probability of surviving. We call that T. And then we have some amount of clonal reproduction C and sexual reproduction F. And we're going to let R sub C be the relative amount of clonal reproduction. It's just the amount of clonal reproduction over the total amount of reproduction. And the way we're going to do selection, this should look a little familiar. We're just going to say that your probability of surviving depends on how far you are from the optimum. The optimum is theta, and you're sitting at z bar. If you're far from theta, your survivorship is very low. If you're really close to theta, your survivorship is really high. So we model it as a little Gaussian fitness function. All right, and then we say, all right, given that, what's the change? So this is before I did recursion, so this is just a difference equation. So here's z bar t plus 1 minus z bar of t. So we can go ahead and do that for both the phenotype and the genotype, and we get, I know they're not so simple, but they're simpler than they were. And these are easy to do to put numbers in and iterate numerically the way we did those equations for the virus. So now we can do something like that, which is what I did here. So here I'm going to make a comparison using those equations from the previous slide. And I'm going to compare with no clonality, so no clonal reproduction, with a case of an arbitrarily chosen value of clonality, because I'm a theoretician. And so here's a graph. This is time. So we're changing. We are following something in time now in sort of steps of time generations. Up here is the genotype and the phenotype. And down here is the population size. Up here, no clonality is the little dotted line. And then genotype and phenotype are straight line and dashy lines. And what I did is I started the population with a phenotype and genotype with value 0, arbitrarily chosen. And the optimum theta is 3. So they suck. They're terrible. They die. They're unfit. But, you know, Darwin was right. Evolution happens. And they evolve. And the interesting thing, and you can sort of see it here, is clonality does not affect where you end up. It doesn't affect the equilibrium. Everybody goes to 3. Everybody goes to the optimum. Selection works. Yay. This is a perfect world, right? So this is a deterministic equation. There's no drift. There's no badness. Everything goes good. So they get there. But what happens is clonality affects the dynamics. It affects how fast you get there and how you get there. So notice that this little dotted line gets there first. So clonality slows down natural selection. That kind of makes sense to us. If you're making copies of your offspring that are exactly like you, natural selection needs variation. And so it's going to work a little slower if there's less variation. All right, so clonality seems to be slowing things down. Doesn't sound great. Doesn't sound so good. But there's something else, and this is not something we would have predicted until we ran the numbers. This is what happens to the population. We started at about 700, and we let it run. And I said at the beginning, we were at 0, and we should have been at 3, so they die, right? It's bad. The population goes boom, down. Eventually, it picks back up. As fitness increases, it picks back up again. Here's the case with no clonality. It bottoms out. It doesn't go extinct because this is the deterministic model. They can't go extinct. They come back up, and they survive. But look at the case with clonality. It doesn't go as far down, and it recovers quicker. Selection is weaker with the clonality. So it gives it this demographic buffer where it allows it to adapt before it bottoms out. So what we're seeing is a demographic advantage at the same time as you're getting sort of an evolutionary disadvantage, which is not what we expected. And this is the idea. Remember I talked about the idea of evolutionary rescue, right? We have to have time to be rescued. If you're way down here, it could be that you just go extinct by random chance before you get your chance to get up here. Even though these guys get up here faster once they make the recovery. But the question is, can you make it through that point? Okay. So this is sort of making that point again. So this is that graph I had before of we have to get out of this demographic decline. And it turns out that what clonality is doing is helping us get out of that demographic decline more quickly. In this specific case of a single change. So I make the point that we use models in 2 big ways. We use it to explain what we do see. So we have specific tests of hypotheses. We take data and we say, can my model explain this data? And if it can, what did I learn about the data? In the case of the dynamics of HIV, we learned that some of what we were seeing with the virions and the cell cycle was implicit. It was a part of the dynamics of basically a predator-prey system. But we can also help us predict what we might see and generate hypotheses. So in this case, it's telling us, hey, we might see a very different response to a sudden adaptive change in an organism that can have some asexual reproduction. And maybe that explains why so many invasive species are asexual. So here's this idea of looking at something where we have data and asking the question, can we predict that? And then having sort of a general prediction of something we don't know about. So I especially like these 2 examples, and I use them sometimes when I talk to just like student groups and stuff about why they should take math classes. And I say, these are really important types of questions, right? These are things we care a lot about. We care a lot about disease and how disease works inside a host and what are the natural dynamics of disease in a host. And we also care a lot about how populations respond to change, whether it's change that we inflict on the environment or changes that are happening in natural systems. So the kinds of questions that we can ask are important questions. All right. I want to say, because I'm important, two of the people who worked a lot on the results that you saw were undergraduate researchers in my lab. Carlos Fernandez is an author on the 2017 paper that we're going to talk about tomorrow. He was an undergraduate in math at Kansas and did a lot of preliminary work on the model that we worked on. Anna Goddard is a current undergraduate in my lab, and she's working on some of the stuff that I didn't present today, but she also works on this with us. But not everybody who works with me is an undergrad. I have collaborators at the University of Florida, some pretty well-known theoreticians, Mike Barfield and Bob Holt, who have been working with me on this. We've been supported by the University of Kansas and the University of Florida. We're on our second NSF grant to do this work. Our undergrads have been supported by IMSD and the KU McNair Scholars Program, and I gratefully acknowledge their support. And then I'd be happy to answer any questions. Thank you for listening. Yes?
You showed this graph where there's the question, is going to make it through the bottom part. The axis showed that you had like a few hundred individuals, and the thing looked perilously close to, I don't know, 10 or 3 or 5. Did you run a individual-based simulation?
Yes, I did.
Actually
yes, I did. I didn't show it here because I thought today's discussion was like the model, the beauty of the model. As soon as we saw those results... Oh, sorry, I'm going to repeat the question. So the question was, I showed this figure, and we had these numbers go down to tiny numbers, and yet the population recovers. Did I run any individual-based simulations or any stochastic type models that would take into account being lost there? And which is a really good question, and sort of of course you should ask that question. Yes, we did. So we did. So this is all a numerical exploration of our deterministic results. We also did fully forward time individual-based simulations. I'll talk about those tomorrow a little bit. In those, we model a single trait. I think it's 10 genes controlling the trait, and we do the full thing, drift and everything. And so for this case, where it's a single change, what we can do is show that the probability of persistence, not the probability, 1 minus the probability of extinction, whichever you want to do, the probability of persistence is better with the clonal case. So you're more likely to make it through. Sometimes you go extinct no matter what, right? But yeah, so there's definitely, and that's to be expected. So as soon as we saw this, we knew that the stochastic model would show an advantage. Again, we get, this is not the phenotype. The phenotype gets there more slowly. So it takes longer to adapt. And so there's cases where that's the important thing. But in a case where there's a sudden change and you can make it, then it's much better to have that little bit of clonal reproduction to buffer you against that sudden change. Yeah, David.
So essentially, it's well known that if you look across the tree of life, that species that only have asexual reproduction and have no sexual reproduction are more prone to extinction.
Right.
Has anybody looked to see if what you're predicting here is true, which is species that have no clonal reproduction have a higher extinction rate than species that have some clonal reproduction?
That's a very good question. So there's a group right now that I'm part of out of mostly French researchers looking at what they're called partial clonality. So organisms that reproduce sexually and asexually, and trying to get a sense of how many or what proportion of them are invasive. How deep are they in the tree of life? How speciose they are. I think that's a really good question. We tend to focus on the extremes. We are completely sexual. We are completely asexual. It turns out there's a lot of organisms that either within the same generation are able to do both, or, like Daphne is the classic example of like things are good and things are bad and we do what we want. And so I don't know. I don't know the answer, but I think that's a really good question. Yes?
Perhaps similarly, I was wondering if you think about changes to your model that would allow for different rates of outcrossing. I think you talked about random mating, presumably outcrossing sex, but what about organisms that are highly selfing?
Yeah, that's a good question. So this model we haven't done any with selfing, I think. I'm pretty sure. I did a different model with one of my former PhD students, Tara Marriage, where we looked at a different version of this sort of thing. And we looked at, we were specifically thinking of a plant model where we had plants that had mixed mating, so they had both selfing and outcrossing, and some amount of vegetative growth. And there's an interesting complex relationship between selfing and asex, because asex freezes heterozygosity, and selfing destroys heterozygosity. So we got all sorts of interesting interactions between them. So I think yes, all of the stuff I showed you today assumed outcrossing, and I think if you allow even a little bit of selfing, things start to get interesting. And I think one of the big things about asex is that there's been, the Michelson, I'm going to say it wrong, Michelson effect, which is this idea of frozen heterozygosity in asexual lineages, and sometimes you see it, but sometimes people look for it and they don't see it. So I'm not, Tanja Schwadlander has a really nice review paper where she's looking at genomes of asexuals. So I guess it's not completely clear to me what the interaction is, but there's definitely a trade-off, right? Because at some point, if you're selfing, also if you're highly selfing, it kind of depends on how often you self and how much heterozygosity you had already, I guess. But yeah, there's an aspect that this is ignoring completely. It's a very good point. Yes?
I really appreciate your interest in connecting math, biology, and kind of breaking down for us how mathematics can be simplified and explained well. How often does it not work so readily?
Oh, it works terribly, many, many times.
And so what's your process? Just can you kind of talk to us about, as a mathematician interested in biology, how you might go about?
So I love to give this, I'm going to give you an example. So I was working on something that I've never published with a dear friend of mine who's really quite brilliant, John Wakely, who's at Harvard. And he and I were working on this model, and we came up with this beautiful coalescent model. We wanted to work on the endosymbionts of sort of marine organisms, and sort of this within and between. We developed this beautiful coalescent backward time model. I showed forward time models. You can always do backward time models. They're really fun. We did that, and the results were really pretty. And then we realized that it was missing a very important biological reality, and it was ridiculous. Like, it was pretty, but it did not capture like something fundamental about the biological process. So we were like, man, we just spent a year writing this really attractive model that is useless. Okay. So then we wrote a forward-time Moran model, which I think is even prettier. It's much better. But so I think a lot of times what you have to ask is, did we miss the important biological thing? And many people have said the important part of a model is including everything you need and not including anything you don't need. So that's where your biological intuition is hopefully helping you. I think a good example of that was actually, I think I forgot to mention it, but thank you. It's like I placed you in the audience for this moment. I have to go back here. I set it up and I forgot it, and you reminded me, so thank you. Notice that this goes up and down. You can't really tell. This model will go to an equilibrium. This is not going to an equilibrium. Something is wrong with that model. When I set up the model, I actually even said it to you guys because I wanted to remember. I said, how are the susceptible cells coming into the system? They are constantly coming in with the immune system. The immune system's constantly putting them in there. That's an assumption that works great for a little while, and then it falls apart. Because what happens in the system is eventually the immune system collapses, and there's no more input of susceptible cells, and then everything goes terribly wrong. So that's a good example of a place where, and that's often true of models, for a range of parameters or a period of time, the model works. But you're leaving something out. You're always leaving something out. In this case, if you leave out the immune system collapse, you get a great prediction of this part, and then but this, you can't explain this without explaining what happens to the immune system. You need a different model to do that. So that's a really great question. Thank you. Yes, I may?
Yeah, I have one question. So, and I'll ask this again tomorrow. What made you interested in this area of research?
Well, I have two stories. I have a sad story and a happy story.
Okay. Sorry,
I'll tell the sad story first, but then there's going to be a happy ending. So I've always really loved math, and I went to Stanford as an undergraduate, and I went to see an unnamed person who was supposed to be an advisor for me. And I went to see this person, and I said, I think I want to be a math major, and I really like math. And this person said to me, I think if you were going to be really good at math, if you were going to be brilliant, we'd know it by now, wouldn't we? And I said, well, maybe. He's like, yeah, most mathematicians are, you'd know. I'm like, okay. So I left, and I was like, right, okay. Well, plan B, I'll take biology. I like biology. I really like biology. I've always liked biology. I love biology. Taking all these biology classes, and now I am going to name a name. I took biostats with Mark Feldman at Stanford, and I loved it. And I went in to see him, his office. I was there recently, and I reminded him of this story. I went to see him, and I was like, Professor Feldman, is there an area of biology that uses math? And his face lit up, and he's like, yes, my area, population genetics. So he gave me a whole bunch of stuff to read. So I blame Mark, and I credit Mark. So it's good because there's these two equally famous professors at Stanford, one who I don't name and one who I do. That's the story. But I always wanted to do something with math, but I love biology, and I love that you can use math this way. And it makes me sad when people don't want to do math. I think it's wonderful. Yeah? Yes?
I wonder if you've used CRISPR or some other methodology to put an impulse function to your model to see how it would react.
To put an impulse into the virus model?
Yeah.
So you can do so many things. So I want to stress that I just picked like a very early predator-prey HIV model. There are complex and very, very good models of HIV. Top-notch epidemiologists have really good models. Not that Phillips wasn't one, but that do explain the system incredibly well. So once you put the immune system collapse in there, you can explain this thing really well. And then the thing that's being done now, which I think is cool, is people are modeling how the antiviral treatments interact with this system. And you can get some very good predictions of using a single antiviral, two antivirals, a combination of antivirals, and doing them in different phases, impulses of drugs and things like that. So I just presented something very simple, but they've gone way beyond this at this point. And I can't keep up with that literature. I've published a little bit in viral. I've published a handful of papers, but I can't keep up with that literature. Those guys they have lots of data, which is the other thing. There's some beautiful work coming out of Plooney Penning's lab that is looking at, you can actually watch the evolution, the phylogenetic evolution of the virus happening in the host, because they have wonderful time series. So her work is really amazing. Yes, you're right here.
Following up on your personal story, what do we need to do to encourage a generation of biologists that aren't math phobic?
So I think about that all the time. So I think the more that we can do to make, I think a lot of them aren't so much math phobic as they don't see the connection. They learn math very isolated from how they learn biology. One hypothesis, and I don't know how, I use a lot of differential equations and things, so calculus was great for me. But one hypothesis is that we should actually be teaching them statistics early on, because statistics is more, yeah, I'm getting a thumbs up from the back. Statistics is more directly tied to biological data and provides a sort of a much better entree into thinking quantitatively about biological systems. I teach an intro biostats class, and I love it. So I don't want us to get rid of calculus completely. We might need it. But But actually, that might be a good thing, because I think if it was clearer how quantitative thinking is important to biology earlier on, I feel like often what happens is you're sitting there, you're taking classes, and then you get to a certain point in like your genetics class, and they throw a bunch of math at you, and then you're like, oh, I didn't see that coming. So that's not right. So I think, I'm not sure because I'm not an expert in pedagogy, but I like this idea. I've had it floated to me a bunch of times, this idea that if we use statistics earlier in our biological studies, the idea of numeracy and thinking about data, because statistics was invented for biology, right? We get it first. So I think it would be good. Oh, and baseball, and baseball. And baseball, obviously baseball. But so maybe that's one idea.
I was a little worried about that. Next time the Royals win and Kansas is home to a world champion again.
We could use the stats. There's a lot of stats in baseball.
I think baseball's a branch of biology, actually.
It's a branch, isn't it a branch of physics? Baseball's physics. Okay. Yes? Did you have a question?
Have you ever done any things where you would look at like nutritional adaptive short-term evolution change, such as dietary, climate, metabolic, inferring to the chemical processes within the cellular structure?
So I've never done that kind of modeling, but I have colleagues who have. So I have a bunch of colleagues that are working on microbiome stuff. And whenever we talk about modeling microbiome interaction and the interaction of the microbiome to fitness of the host, and also the microbiome affects the fitness of the host, but the host is also the environment in which the microbiome interacts with itself and other species. That's where understanding, a better understanding of sort of physiology, basically, nutrition and all those things. I feel like we're sort of talking at cross purposes a lot, the people who model the dynamics and the people who actually understand what's happening at a cellular level. But I think that's an area where I think that conversation is starting to happen. Because you can look at perturbations, right? Jaime and I were talking about this. We were talking about oxygen in the gut.
Oh,
Right? So how constant is a gut microbiome? And I don't know the answer to that question, actually. And I think that's the kind of question that a modeler needs to know, right? I need to know if, do my dynamics change over time? And do some things. And so there's a lot of information. If I was going to do a good job modeling that system, I would have to have a lot more detailed biological information that I don't have yet. Well, thank you guys. That was fun.
And before you all take off, I just wanted to remind you that if you're interested in learning more about Dr. Orive's work, we will have a second presentation tomorrow at 12:30 in the marquee at Union South. It's a little more technical, but it's open to the public. Yes.
It's so fun, I promise.
Yes. Thank you all for coming.
Hope to see you next week for the first talk.